Welcome participants to the fourth webinar organized by ISAAA Inc. in collaboration with Winrock International, the implementer of Be Safe or Building Safe Agricultural Food Practices, Food Enterprises project. Today's webinar is Biotechnology Applications and Impact on the Philippine Livestock Industry. I am Dr. Rhodora Romero-Aldemita. You can call me Ola, the Executive Director of ISAAA Inc. in the Philippines, your moderator for today's webinar. Currently, the Philippines is completing the final draft of Animal Biotechnology Regulation after a series of consultations with regulators, scientists, and biosafety committees. Meanwhile, research and development on animal, especially livestock, our topic for today, livestock biotechnology being conducted by a few research institutions such as the Philippine Carabao Center with the Livestock Biotechnology Center and the academe such as the Central Luzon State University, University of the Philippines, Los Baños, and some few others. There are enormous potential benefits, of course, of animal biotechnology and its products for food, agriculture, animal health, and welfare. So information on the Joint Department Circular 1 for Animal Biotechnology, the benefits of the technology for the livestock industry should be disseminated to all stakeholders to guide researchers, importers, and food producers. Today, Be Safe, ISAAA Incorporated, and through our experts, we aim to encourage acceptance of livestock biotechnology in the Philippines and to highlight the current opportunities and potential benefits of trait improvements related to food and agriculture and resilience to climate change, most importantly. So this webinar is a part of a series of webinars on an array of topics about modern agri-biotechnology applications and products in the Philippine context. Now let us share the house rules, the slide for the house rules, please. So the webinar will run for 2 hours. We plan to have it for 1.5 hours, but if there is really very good discussion, we can run it until 2 hours with lectures, all questions, and discussion. All microphones are on mute and questions should be entered in the Q&A function only, please. The chat function will be used by the panelists and for important messages to the participants. All the presentations and the Zoom recording will be posted at the ISAAA Inc. website and freely downloadable. So it's at ISAAA.org. That will be posted after the webinar. We also invite you to stay till the end of the webinar and respond to a post-webinar survey, and respondents will be given certificates. And I'd like to stress the importance of the poll questions. They will be introduced every after speaker. Okay, so to formally start the webinar, let me now introduce Dr. Claro Mingala, the Director of the Philippine Agriculture and Fisheries Biotechnology Program, Department of Agriculture. He was the former Center Chief of the Livestock Biotech Center at the Philippine Carabao Center and the Chair of the Technical Working Group for the drafting of the Joint Department Circular for Genetically Modified Animals and Animal Product Rules and Regulations. Friends and colleagues, let's welcome Dr. Claro Mingala to give the opening message.
Hello, thank you very much for the kind introduction, Dr. Ola. And thank you very much for this opportunity for always inviting us in behalf of the Department of Agriculture, Biotechnology Program Office, and of course, in behalf of our steering committee chair, Undersecretary Mercedita Sombilia, of course, of the Department of Agriculture. Good morning to our organizers, speakers, and our audience. I would like to thank, as usual, ISAAA, headed by Dr. Aldemita, the USDA, the Winrock International, through the Be Safe project for organizing this webinar. Biotechnology application in the livestock sector is a very timely topic considering its potential to address global food security and sustainability challenges. Among the most advanced technology tools in livestock research and development activities includes genetic engineering, gene editing. These techniques are now used for genetic improvement, disease prevention and control, and improving animal welfare. Genetic improvement can include breeding animals with desired traits such as increased milk or meat production or resistance to certain diseases. Disease prevention and control can involve the development of vaccines and diagnostic tools, as well as improving animal resistance to specific pathogens. Improving animal welfare can include reducing stress, improving nutrition, and developing more humane methods of production. These activities can have significant benefits for farmers, consumers, and the environment as well. There is also growing interest in using animals for production of bioproducts, such as enzymes, hormones, and antibodies. This can be done through genetic engineering or other biotechnology techniques and has the potential to provide new sources of income for farmers and new products for the industry. Global opportunities in animal biotechnology in livestock are significant, and research and development in this area are likely to continue to grow in the coming years. I find the topics to be discussed interesting, and so I would like to encourage everyone to actively partake in this webinar and take advantage of the opportunity to engage with our resource speaker. So thank you very much again, and we are expecting that more information and all about livestock biotechnology or animal biotechnology would be impart to our participants in today's activity. Mabuhay po tayong lahat. Good morning.
Thank you so much for that inspiration. And we know that as the number of attendees increases, we see that the interest in all our participants today. So we're very happy and honored to have our first speaker. She is the Cooperative Extension Specialist in Animal Genomics and Biotechnology at the Department of Animal Science, University of California, Davis, USA. She conducts publicly funded research and outreach program that focuses on the use of animal genomics and biotechnology in livestock production systems. Her current research projects include the development of genome editing approaches for cattle, and she has been a recipient of the CAS 2014 Borlaug and the ISAAA 2019 Rockefeller Prentice Award in Animal Breeding and Genomics. Colleagues, let us welcome Dr. Alison Van Eenennaam for her talk.
Thank you, Dr. Ola. You can hear me well?
Yes, very well. Thank you.
Okay, then I'm going to go ahead and share my screen, and then I will start speaking to you. So, all right, you're getting a full screen there? Thumbs up?
Yes, please. Yes, we have.
Okay. All right. So, all right. Well, it's great to see so many people interested in livestock improvement. Obviously, that's my life's work. I'm at the University of California and have been here for 20 years and have really lived through both genetic engineering and now genome editing. And so my objective this morning is to talk about the kind of the background of breeding. For those of you that don't know too much about livestock breeding, we hear a lot about plant breeding, but perhaps people aren't aware of what we can do with livestock. And perhaps you have one of these at home and you can appreciate the changes we can make to animals based on selecting for a given appearance that we consider to be an improvement. Maybe the wolf that these dogs are derived from don't think it's an improvement, but clearly we can make tremendous genetic changes. And when we're talking about these kind of changes and we're looking at things like milk production or yield, it's really quite stunning the types of changes that we've made over time. So this shows milk production in the US, 1944 through to 2019. And on the left axis over here is the number of animals. And so back in the 1940s, there were about 26 million dairy cows in the United States. And then what happened was genetic improvement began, and we started to select for healthier, more fertile, more long-lived, more productive animals. And what we see is that that enabled a real drop in the number of animals, so that today we have around about 9 million dairy cows, but we're producing about twice as much milk with that very much reduced herd as a result of the fact that each cow is producing much more than she did in the 1940s. And the reason that is a contributor to sustainability questions is shown in this slide here, which compares 1944 levels shown here at 100%. Today's levels shows that milk production per cow is over 4 times what it was in the 1940s. And as a result, we're using only 21% of the animals, 23% of the feed, 35% of the water, 10% of the land, 24% of the manure, and ultimately the carbon footprint of a glass of milk today is around about 1/3 what it was in the 1940s. That's the sustainability contribution of improved genetics. And I think a really important point that a lot of people don't know about, it's been happening just using conventional breeding. And this kind of summarizes what we might modify animals for and the types of tools we might use to do it. So we've used selective breeding, we've used artificial insemination, embryo transfer, genomic selection, all of these to improve our animals. And today's topic is really about these modern biotechnologies. So specifically cloning, genetic engineering, and genome editing. And for me as a geneticist, I see these all as breeding tools, and I don't draw a bright line in the sand when I start using molecular tools. But clearly that has been the case in terms of at least regulation around genetic engineering and maybe genome editing. Today, I'm going to focus on agriculture and food production, that is modification of animals to produce food. There are other reasons you might modify animals for things like pest control and control of malaria or that type of thing. You might make fluorescent fish because they look pretty cool, like the glowfish here, which have a transgenic DNA that expresses fluorescent genes from jellyfish. You might produce pharmaceutical drugs in goats and chickens and cows. You might produce pigs to produce hearts that can be transplanted into humans. You might do research on the literally billions of genetically engineered mice. But today I want to focus on agriculture because that is really where the discussion around food sustainability lies. And so I'm going to talk about some applications that exist in agriculture. And this is kind of an overview of biotechnology, and by that I mean modern biotechnology's use in animal agriculture. And so the very first transgenic animals were produced back in 1985. And that was done with some pigs and sheep. And then in 1989, there was a very famous fish, the Aquavantage salmon, was first produced up at a public university in Canada. Herman the bull was produced. And then we had, of course, the infamous Dolly that was cloned in 1998. We had the fluorescent glowfish, we had fluorescent chickens, we had myostatin knockout cattle. And then in 2010, we started having the introduction of genome editing reagents like zinc fingers, talons, and CRISPRs to produce the next generation of biotechnology animals. And so that kind of gives you an overview of what's happened. So this technology has been around actually for quite a reasonable amount of time now. But I think if we compare it to the types of global adoption of genetically engineered crops, we see a very different picture. And that is, at the current time, there's only 2 approved genetically engineered animals for food production. That is the Aqua Advantage salmon, which, as I mentioned earlier, was first produced in 1992. And then there was a GelSafe pig, which has had a protein inactivated that produces...
For centuries,
which produces a protein that results in an allergy from people that have red meat allergy. And so at the current time, these are the only 2 things that have ever been approved. And actually, this product was only relatively recently available for commercial sale. It is now available in the US, but it was actually first available and approved for commercial sale in Canada. And that was a couple of years ago now. And so it's been a very long road to market to get these products through to commercialization. So that's genetic engineering, kind of old school molecular genetics, if you will, introducing a transgenic construct, for example, a growth hormone from Pacific salmon into Atlantic salmon, or using a construct to inactivate a gene. And that's quite different to the kind of newer methods of genome editing. And so I'm going to play a video that hopefully you'll be able to hear that explains a little bit about what it is that we can do with genome editing. And so I'm going to give my computer a minute to load up this video, and it will go over in a very kind of diagrammatic and simple way how genome editing works, and hopefully you'll see how it differs from genetic engineering, because we're not necessarily introducing a transgene or basically a sequence of DNA from a different species as we were with genetic engineering. And I'm not having my slide advanced. Let's see if I can get it to work this way.
For centuries, animal breeders have selected individuals displaying desirable qualities and bred them to produce particular goals. Breeders are actually selecting unnaturally occurring DNA sequence variations that result in differences between individuals, like the color of a cow's coat. Using conventional breeding programs, achieving desired changes can take decades, especially in large livestock species like cattle, which have long generation intervals. Innovations in biotechnology are poised to help breeders improve the rate of genetic gain. University of California, Davis, animal geneticist Dr. Alison Van Eenennaam and members of her laboratory are exploring genome editing tools that could allow breeders to precisely introduce useful genetic variations into livestock breeding programs. Genomes are made up of DNA, containing the collection of genes, unique instructions detailing every characteristic of an individual, what they look like, the amount of milk produced, or resistance to disease. Genome editing offers an opportunity to edit these instructions precisely. One method to edit genomes is the CRISPR-Cas9 system. This system has 2 components: the Cas9 protein, which cuts DNA, and a guide molecule that directs Cas9 where to cut. Bound together, they form a complex that introduces a precise cut in the DNA. The break is then mended by the cell's natural DNA repair machinery. This can be used to inactivate undesired genes, like those that cause disease. Alternatively, the repair machinery can introduce useful DNA sequences which result in desirable traits, like heat tolerance. Genome editing complements traditional breeding methods. It has the potential to introduce useful traits like disease resistance and adaptability into genetic improvement programs, addressing some of the problems facing global agriculture.
So hopefully that was an explanation of how genome editing works. And what I would like to go through in the next few slides is where we might use genome editing in our our breeding programs. I'm sorry, my screen has just frozen and this is not the slide I want it to be on. Okay. Oh gosh. Let me see. I'm going to jump through that. Sorry, it's this thing is, there we go. All right, there we go. So what might we inactivate using genome editing? So the very first example was just using the editor to go in and inactivate a gene, that is turn it off. And basically, as was alluded to earlier, the types of things we might want to inactivate will be things that make animals susceptible to disease. We might want to remove allergens. We might want to remove unwanted development. We might want to make them more resistant to heat or thermotolerant, and we might want to increase yield. So let's have a look at some examples. And the flags represent where research in this is going on, and it's really based on a summary of a conference I attended earlier this year with Dr. Ola in Brazil, where we kind of heard an update from the global community as to what they're working on. So one of the topics was inactivating a protein that results in a virus that can attack pigs called porcine reproductive and respiratory syndrome virus. And basically inactivating this gene means that those animals are no longer susceptible to that virus. And the areas shown in orange on this slide are basically all the countries where that disease is a really prevalent problem. And so there's work in Scotland at the Roslin Institute, in China and in the US at University of Missouri to basically inactivate that gene and make pigs aren't susceptible to that disease. And I guess to me, as a geneticist, I think making animals resistant to disease is actually better than having animals get sick and then having to either treat them with antibiotics or chemicals or perhaps even have them die, which would be the most unsustainable of all the options. There's work in New Zealand to inactivate the gene called beta-lactoglobulin, which is one of the major allergens in milk. So you can go in and turn that gene off so that protein is no longer produced in the milk. So the milk would no longer be allergenic to people who are allergic to beta-lactoglobulin. This is work from the US with Excelogen, who are working on a welfare trait of pig castration. So the male pigs, as they grow older, get a thing called boar taint, which makes the meat have an unpleasant taste, and the animals get aggressive. So they're doing work, and this just got published, to generate pigs with a knockout in a gene that is associated with development going to sexual maturity. So those pigs stay effectively adolescent and don't ever develop boar taint. So that's an animal welfare trait. Another example here is a gene called prolactin receptor, and this results in a knockout called slick, which gives these cattle a very slick hair coat, a shiny, sheeny hair coat compared to wild type. And on average, the body temperature of these animals stays about 1 degree lower than animals that don't have that knockout. And this is a naturally occurring genetic variation that occurs in Senapol, for example, some tropically adapted breeds of cattle that you can now introduce potentially into animals that aren't necessarily heat adapted to help them cope with a rising temperature. There's also work being done in China on a number of different fronts. There's work being done in all three of these species, so pigs, cattle, and sheep, to inactivate myostatin, which is a really important regulator of muscle development. And if you inactivate that gene, you get about 30% increase in muscle yield in the animals. So they produce more meat. And you can see in the images down here that the animals that have the myostatin knockout have this kind of more muscly appearance. And it actually mimics naturally occurring variation that is seen, for example, in quite a lot of European breeds, as is shown here. This is a picture I took in October at a show in France, naturally occurring double myostatin knockout, resulting in this very, very muscly appearance in this cow that was at this show. And there are also groups that have worked to knock out myostatin in fish. And in fact, there have been myostatin knockouts in Japan of the pufferfish and the red sea bream, shown over here, that are actually commercially available on the market. And so that's the first gene-edited animal product on the market in Japan. And they have gone through their regulatory and they've decided they're no different to conventional breeding, because that is how myostatin knockouts occur. And similarly, Brazil and Argentina are of the opinion that if there's no novel DNA introduced, then they're not going to be treated as a quote unquote GMO. And so a similar approach for a tilapia was given a non-GMO status, although those animals are not yet being commercially for sale in Brazil and Argentina. So those are some examples of genes we might knock out. What might we knock in? So with that pink DNA, the template repair template you saw with homology-directed repair, and basically nothing changes. We still want disease-resistant animals. We want animals that don't have unwanted development. We'd like them to be thermotolerant. We'd like them to have improved food quality and nutrition. And so we do edits that can actually help with these types of breeding objectives. And so I'll give you another few examples from around the world. Work in China, working on tuberculosis resistance. Tuberculosis, of course, is a zoonotic disease that can transfer between humans and people and is a global health concern. And then this was an example from China that was shown at this conference in Brazil, where they're actually doing a triple edit. So they're editing pigs to not only be myostatin knockouts, so very muscly, but also to be resistant to PRRS. And then thirdly, to be resistant to transmissible gastroenteritis virus. So a triple edit to make more productive, healthier animals that will produce more meat per unit of pig. And that's work out of China. And there was actually, according to the researcher that gave this talk, Dr. Q, around 20 gene-edited livestock examples that are in the early stage of development and safety evaluation in China. And 3 of them are at the kind of stage of getting production tested. So that's very exciting. And some of the things that has been worked on there are pigs that are resistant from classical swine fever virus. And some of you may be familiar with the perhaps much more devastating African swine fever, which has been reported throughout the world and spreading. And I know that it spread from Europe over to China, and it spread throughout Asia. And Australia is very nervous right now because that's a very, very devastating disease. And it will be wonderful to have a gene-edited or even a genetically engineered pig that was able to survive that virus. Here's more work from New Zealand, where they've worked on a gene that changes black and white Holstein cattle to this more pale brown and white animal that would not absorb heat as much. That's a coat color one. And then one of the projects that I've been associated with in collaboration with Accelergen is bringing the trait of not growing horns into dairy cattle breeds, which typically do grow horns. And you may or may not know that normally they're physically removed. So they're either burnt off or cut off as the animal gets older. And there's a simple 212 base pair duplication that results in this dominant allele at the pole gene. It's a naturally occurring allele that if you bring that into the recessive horned allele, you'll result in animals that don't grow horns. And they produced a bull that was carrying this dominant, what's called the poled allele, that resulted in offspring that we produced at Davis, where we crossed that bull to some horned animals, and the offspring from the edited bull didn't grow horns. And so this is Princess alongside her control that was produced by a Holstein that wasn't edited. And you can see that Princess there didn't grow horns, but her control, Cinderella, did. And similarly, these are some other animals from that bull alongside their controls. And so this is an animal welfare trait. And we've actually been working for a number of years following these animals. We just recently published a paper looking at the milk and meat and growth and health of these animals from birth to death, and are happy to report that there was no difference in any of those parameters as it was compared to animals that resulted from a bull that hadn't been genome edited. And as you would perhaps expect, because this is a naturally occurring allele that we see in our beef cattle populations routinely. So in summary, I think that this technology and genetic improvement in general is a really important driver of agricultural sustainability. Biotechnology offers an approach to introduce this useful variations like not growing horns or disease resistance or different product improvement without having to do a crossbreeding, that is mate an animal to another animal. You can just bring in the allele from the gene you want without the rest of the genomics of, for example, a beef animal being introduced into dairy cattle genetics. Traits that have been targeted include disease resistance, heat tolerance, growth, and animal welfare traits that sometimes are very difficult to address using conventional breeding because you don't want to bring in all that extra genetic baggage from, for example, beef animals into dairy animals. And really, to me, the fate of this technology will very much depend upon developing some sort of harmonization and regulatory framework that enables this technology to be used, and more as importantly, enables international trade of products like milk, meat, and eggs from these animals, and additionally, gametes, sperm and oocytes and embryos, because the livestock breeding industry is a global industry and we import genetics from all over the world. And so there needs to be an easy way for that to occur. So I thank you for the opportunity to speak to you and just want to acknowledge the funding sources of my lab, and I look forward to questions. So with that, I'll stop sharing and pass it back to Dr. Ola.
Thank you so much, Dr. Alison. Alison, yes, we remember that that 1 week in Brazil where we were really fed with a lot of information. It was so much, and we appreciate really your participation in today's webinar. And then we can now proceed with the poll questions. Thank you so much, Alison. You can also, as we put on the polls, you can also help me in getting the correct answers, if it's okay. All right, so here are the poll questions. We invite everyone to respond. I am seeing people logging in and answering the questions. Please respond to the poll. So there are 3 questions. The first, transgenic livestock were produced in what year? And then the second one is, which of these is not a gene editing reagent? And the third question is, what was the first genetically engineered animal product commercialized for food use globally? We heard that very well today. And what was the first gene-edited animal biotech product commercialized for use globally? Okay, so we're still waiting for all your responses. And here we can see how much attention you gave to Dr. Al's talk.
There are a couple of raised hands. I'm not sure if you wanted to allow those people to talk.
Not yet though.
Okay, all right.
At the end. Yes, when we have the Q&A.
Okay.
Okay, how many more minutes do we wait?
Okay. It looks like about 38 have logged in, so give them a couple of seconds to work out how to use the poll machine. I know I personally can't use it because I guess I'm a presenter, so.
But you can see the responses, right?
I can, yes.
Yes, okay. So when the end of the poll is there, when we reach that, then we can discuss the response.
Okay.
Yeah.
You tell me when you want me to do that.
All right. So 100% has
we've got 60 responses so far. Okay, everyone, we have to get up to 100 responses. So if you haven't responded, you need to respond now, and and then we'll close the poll. Does that sound reasonable? And if it doesn't happen quickly, I'll close it anyway. So this is your last minute to respond. Come on, I gave you the answer to every one of those questions in my presentation. So far, you guys are getting 3 of them right, but you're getting 1 of them wrong. So we'll see if we pull out of that.
Oh, okay, yeah. Are we there yet?
We're at 76.
We have 167 participants.
I know some people are very shy. You know, I can't see who answered what. I'm not going to like call you out and say that so-and-so got question 2 wrong. I really can't see that. But I will tell you that you're getting 3 of them right and 1 of them wrong. So
how many minutes did we allow for the poll, please? Should we stop? Have we had 1 minute yet? Okay, so 3 more seconds.
3 more seconds. All right, we're at 90. Come on, everyone, you can do it. 92, I see.
Okay. Very interesting responses. Okay. For all those who have questions, please type your questions in the Q&A box. And we will deal with that at the Q&A session later on. Okay, I think we can close this poll session now.
Oh, we got to 100. Yay. Yep, we're good. We got to 100.
Yes. Okay, can you see it, Alison?
Yes, I can. I'm going to share the results. Am I sharing the results?
You can read the results.
Can you see the results?
Yes, yes.
Okay, so the first transgenic livestock were produced in 1985. So you guys got that right. I put 75 just to see if people were paying attention, but 1985 was the first transgenic livestock. There were some earlier transgenic mice, but livestock was at 1985. Which of these is not a gene editing reagent? Taq polymerase is not a gene editing reagent. So well done there. And then first genetically engineered animal product commercialized for food use globally was the Aqua Advantage salmon. So the glowfish preceded it, but it was not a food animal, or at least it shouldn't have been a food animal. EnviroPig was never commercialized, and the GelSafe pig was only approved two years ago. And so the Aqua Advantage salmon was the first. And bonus points if you know which country or economy. That was in Canada, was the first approval. Then, which was the first gene-edited animal product commercialized for food use? And this is the one you got wrong. So it was the fish. So the knockout fish in in Japan. So there was the knockout puffer fish and the red sea bream. And that was approved, I believe, in last year and is currently commercially available in Japan. And that's actually the only approved products that are from a genome edited animal. So the fish and Japan was the correct answer to that.
Thank you so much. Thank you so much, Alison. You may please wait a while. We'll proceed with the second speaker. Thank you. Let's give Alison a round of applause, a virtual round of applause for the meantime. Okay, so let's now move on to our next speaker. Dr. Marvin Villanueva is the director of the Livestock Biotechnology Center, and his areas of expertise include food safety, antimicrobial resistance, adulterants, and veterinary drug residues in food, livestock biotechnologies, epidemiology, and addressing operational health problems in ruminant farming in the Philippines. He is a recipient of the 2021 Outstanding Young Scientist Award in the field of veterinary medicine, so he's really still young. And the 2018 PSAS Outstanding Young Professional in Veterinary Animal Science Research. Colleagues, let us welcome Dr. Marvin Villanueva.
Thank you very much, Dr. Ola, for the kind introduction. Let me just share my screen. Am I audible?
Yeah, you are, but still a little bit muffled, but yeah, we can hear you.
Okay.
Yeah.
Okay. So again, good morning to everyone, and it's really an honor to be part to be one of the speaker for today's session. So My topic is about opportunities for livestock biotechnology in livestock and its impact on the industry in the Philippines. So while listening to the presentation of Alison, it's good that some of the products of the livestock biotechnology developed by several researchers all over the world were already discussed. Actually, some of those are also included in my presentation, but I will just give some brief introduction to it. So since I will be talking more on the problems that the Philippine livestock industry is facing, so I will try to bridge what are those opportunities that animal biotechnology as a tool can be used in providing a food secure and food sustainable Philippines. Okay, so let's start. So the livestock industry in the Philippines is continuously faced by several problems nowadays. Those are the transboundary animal diseases. It is exemplified by the African swine fever, which is until now is continuously devastating the livestock industry, the avian flu, and other diseases. Another is the high cost of farm inputs, wherein those raw materials such, for feeds such as soybean and corn, which are mainly imported from other countries, shoot up the prices. That's why as of this moment, the price of the commercial feeds are very high. Another is the lack of new breeding animals, as what mentioned by Alison. There are many techniques that we can employ using modern biotechnology techniques. So in the Philippines, we do have native animals. And maybe one example is the native Sikihor cattle, which has a very good milk production. But if we talk about the if we talk about feeding 109 million Filipinos and more, we really need to have a very good animals that has higher production capacity. Most of the time, we usually sought imported animals, but it is not sustainable, and we really need to be very careful, especially on the biosecurity measures as a practicing veterinarian, so that the entry of those diseases will not go here in the Philippines. And then lastly is the negative effects of COVID-19 pandemic. So around 3 years ago, the supply of the livestock products were sometimes hampered because of the travel restrictions all over the country. Good thing that slowly it is getting better as we are slowly transitioning to our old normal situation. So these are just some of the major problems that the industry is facing. Actually, there are more, but I'm just focusing more on this. Okay, so this is the graph that I got from the USDA Foreign Agricultural Service about the Philippine livestock and poultry update. So I just like to exemplify the effect of the African swine fever in the swine industry. As you can see in this graph, the supply is really going down thanks to the importation, wherein somehow it helps meet the demand of the Filipinos. And really, the swine industry is hesitant to rebuild stock despite the government's best effort to encourage repopulation amidst the continuous threat of ASF infection and in the absence of the locally available vaccine. So as maybe you can see from the newspaper that there are several collaboration between countries like Vietnam on the use of vaccine, but until now it's still there are still gray area, or we cannot see yet good results from these vaccines from other countries. This is another picture or status of the African swine fever zoning status as of May 2022. As you can see on the left map, still the ASF is still devastating the whole country. A good thing for those who are those islands that are separated from those connecting islands, they were able to maintain some free zones. And on the right graph, or on the right map, you can see that the number of days with no reported cases are increasing. So I think this is through the efforts of the national government as well as the local government units and other stakeholders who are working closely to prevent or to reduce the ASF cases in the Philippines. So with the current problems that we are facing at hand, so here comes the opportunity that biotechnology has to offer, particularly in the livestock industry sector. So first, let me just define what is biotechnology. So this is the art or set of techniques or a tool of utilizing living creatures and their products for food, drink, medicine production, or for other benefits for human and other animals. So it was discovered that the animal biotechnology has been practiced a long time ago. So I saw from the presentation of Alison that the year 1985 where the transgenic mice developed or created, but as early as 18,000 years ago. So this is just the traditional animal biotechnology, wherein it was practiced such as domestication and artificial selection was given for a given animal. And as you can see here on the lower left photo, you can see the increase of weight of a broiler chicken in 56 days of management. Actually, right now it's just 31 to 32 days, which is a product of biotechnology. And the modern animal biotechnology, as what Alison mentioned a while ago, began only following the discovery of the genetic code. And some of these products of modern animal biotechnology includes the CRISPR-Cas system and the gene editing technology. Like that. So other traditional biotechnology techniques also include the artificial insemination and also the development of vaccines. Okay, so the following are the major benefits or positive impacts of animal biotechnology as a tool in attaining food sufficiency and security in certain country or community. So that includes the development of climate change, and this is resilient or resistant animals. Increased income for livestock food producers, doubling food production to meet the supply for demand, proper animal management, since as what Alison mentioned, the one study from the China, they try to infuse three traits or they try to address three problems using in one animal. So that's very good. It's really difficult to take care of animals with so many problems, be it the health, the production, etc. And the last one is the rapid diagnosis and modern disease surveillance. So let me just mention about the Cartagena Protocol on biosafety the Convention of Biological Diversity. So it is a international treaty governing the movements of living modified animals resulting from modern biotechnology from one country to another. So this is adopted on the January 2020 as a supplementary agreement to the Convention on the Biological Diversity and entered into force on September 2023. So let me just give you some background since I think not most of us are familiar with this. Okay, so let me just give you some terminologies which we will come across in several slides. So first is the living modified organism is any living organism that possesses a novel combination of genetic material obtained through the use of modern biotechnology. And then the living organism is any biological entity capable of transferring or replicating genetic material, including sterile organisms, viruses, or viroids. And modern biotechnology means the application of in vitro nucleic acid techniques, such as the DNA or direct injection of the nucleic acid into cells or organelles, or the fusion of cells beyond the taxonomic family that overcome natural physiological, reproductive, or combination barriers, and that are not techniques used in traditional breeding or breeding and selection. Okay, so there's also another term, this genetically modified or GM animals. It involves altering its genetic material by adding, changing, or removing certain DNA sequences in a way that does not occur naturally. It aims to modify specific characteristics of an animal or introduce a new trait such as disease resistance and or enhance growth. So there are two methods that that can be used: the transgenesis or cisgenesis, or transfer of genes in animal, and deletion of genetic information. So for the transgenesis or cisgenesis, so it is a transgenic animal are those which have been there's a deliberate modification of its genome, the genetic makeup of an organism responsible for the inherited characteristics. So as you can see on this simple illustration, while the other one is just knocking out, just like Alison mentioned, knocking out or knocking in. Okay, so these are some of the thematic areas and several techniques that can be performed or done through modern animal biotechnology under the inputs and the uses or outputs of these modern technologies in the field of biomedical and agriculture. So I will discuss some of examples of these techniques that are already practiced by some of our groups here in the Philippines, and those that can be done as an opportunity for these animal biotechniques to be used as a tool and be helpful in our Philippine livestock industry. Okay, so these are just a general strokes that can be used for the new breeding innovation as a driver of change in livestock. So as what the first presenter mentioned, so there are some modern biotechnology techniques like the CRISPR-Cas system. This is the most famous gene editing tool that we can use, and we can do the cell manipulation or micro manipulation of zygote, and then embryo transfer, and then next is the application in agricultural and biomedical areas. So CRISPR-Cas system is a very powerful tool that the researchers can use to replace those unwanted set of gene and replace it with desired gene of interest. So this was already mentioned a while ago, the polled cattle in here and the muscling cattle. Next is animal cloning. Dali is a best example of this, and I think this biotechnology technique has already been practiced here in the Philippines. And since I am here at the Philippine Carabao Center, so the somatic cell nuclear transfer technology has been introduced or practiced here in the Philippines. And we already have successful offspring that was produced out of this clone, not only in water buffalo but also in cattle. So another technologies that we can use is the GM mosquitoes. This is actually the product of the Oxitec, wherein those genetically engineered male mosquitoes prevents the proliferation of female mosquitoes. Thus, it reduces those mosquitoes that carry diseases such as malaria, dengue, etc. And the sleek cattle was also mentioned. The ASF vaccine, which are still in progress. And then the PRRS resistant pigs and the bird flu resistant chicken, which are both products from the Roslin Institute by the University of Edinburgh. Okay, so another application of this genetically modified animals are these GM animals that can be used for disease models, wherein, for example, the HIV is only it can only replicate in chimpanzees, but there is already a way to for this HIV to be used as disease model for macaques. And another is the transgenic mice that can be used for test system development, like for example in poliovirus. So this work are still in progress in developing new models by altering the susceptibility of mice to pathogens of humans. Another is gene therapy, wherein this functioning gene is inserted so that it can be used for example, in cystic fibrosis in this example. Another is xenotransplantation, wherein maybe we can see from the internet that this heart or kidney from the genetically engineered animals like pigs can be used as a replacement of the heart or kidney from humans. So they really prefer larger species such as pigs and baboons to be used as a source of organs for this animal. And then next is the insertion of the genetic material such as from excretion of biopharmaceuticals in milk, protein expression to alter meat composition, and also transgenic mice. So let me just give you some broad strokes on the status of the GM animals regulatory policy. So right now, as what Dr. Ola mentioned, through the efforts of the Bureau of Animal Industry Biotechnology team, headed by Dr. Pingala and uh, uh, Dr. Halos, the necessity to establish guidelines for GM animals and animal products was already recognized. And just like the JDC or the Joint Department Circular that was developed for plants and plant products, this proposed guidelines for animals and animal products also involves different agencies such as DOST, Department of Agriculture, DENR, DOH, and the DILG. So this proposed guidelines is applicable for the following, such as the genetically modified fisheries and other aquatic resources, domesticated animals and biological products used for animal husbandry or veterinary purposes, and lastly, biological agents used for biocontrol derived from the use of modern biotechnology and containing novel combinations of genetic material. So it is also emphasized in this JDC that the proposed guidelines that the products of gene editing do not contain novel combinations of genetic materials are not covered by this JDC. Okay, so I think this is my last slide as a parting words. It is very important to have a harmonious collaboration between different stakeholders such as the government agencies, state universities and colleges, whether it is from the local or in the international areas, especially those international universities that are more capable of doing this modern biotechnological techniques, the local government units, and also some private stakeholders who are interested in pursuing this kind of activities. Also, let us not forget the enabling environment backed by sound policies on GM animals and funding support either from local or international funding source. To have this modern biotechnology tools be maximized, or we can take advantage of these benefits so that we can really help the livestock industry and the community in general to have a food secure and sustainable Philippines. So I think that ends my presentation, and thank you very much for listening, and I'm happy to entertain your questions.
Thank you so much, Dr. Marvin Villanueva. Let me now proceed with our poll for this presentation. Floor, there you go. So we may now respond to the questions. So there are again 4 questions. So the following are the positive impacts, benefits of animal biotechnology. So yes or no, doubling, increased income, doubling food production, proper animal management, rapid diagnosis, and climate change. Okay, the second question is, transgenesis and cisgenesis are the 2 methods in developing genetically modified animals. Okay, please respond. And then the third would be somatic cell nuclear transfer has been adopted and applied in the Philippines. 4, products of gene editing that contain novel combinations of genetic materials are not covered by the JDC1. Okay, so these are a little bit catchy. So let's have a minute of this, and then Dr. Marvin can help us in reading and the responses and say which one is the correct answer. All right, have we gotten already a minute?
I think we should also wait for 100, just like Alison.
Oh yes.
So right now, right now there's
Yeah.
already 86 respondents. So
Okay. Yes, that's a good one.
okay. I
Nice.
didn't make too much difficult questions.
Yeah. So while waiting, let us invite everyone to please put your questions in the Q&A box, Q&A function. We'll deal with them after this poll question.
Okay.
Okay.
We
So you
already
have
reached
102.
102 participants. Yeah.
And we stop the poll now, and Dr. Marvin will read it.
Okay, for the number one, it's yes. I included all of those positive impacts and benefits of animal biotechnology. So majority got the right answer. And then for the number two question, actually transgenesis and cisgenesis are just the same. So it's false.
Yes, true.
Yeah. And then number three, somatic cell nuclear transfer has been adopted. It's true. It's already practiced here in the Philippines. Actually, the Philippine Carabao Center is doing it in collaboration with other partners.
Mhm.
And number four, products of gene editing that contain novel combinations of genetic materials are not covered. So it's false. Not the answer is products of gene editing that do not contain novel combinations of genetic materials are not covered. So I mentioned it in the presentation.
Okay, thank you so much for the responses and also for Dr. Villanueva. Let's give him a virtual round of applause again. And now we continue on with our webinar with the Q&A. Can you spotlight all of us? And at this point, we have received a lot of questions that Dr. Alison has already responded. So may I ask Dr. Alison can we go back to those questions and read your answers, if you please? Because our participants do not know those questions and the responses as well. So maybe we'll start with Nida Alejandro. How about the quality of the meat of genetically modified animals?
Sure. So that's a bit of a trick question because it depends what they've been genetically modified for, right? So if you genetically modify them to change their horns, that's not going to affect their meat production. But those big old muscly myostatin animals, the cattle, their meat is very lean. It doesn't have very much fat, but that's because myostatin's been knocked out. So the naturally occurring myostatin meat is very lean also. It's not because they were genome edited, it's because the trait you inactivated makes the meat lean. So I think it depends what the animal is as to what the impact might be, but it doesn't make the meat unsafe.
All right, thank you. And the next question is from Renny Boy Laconsay. Can we produce meat in vitro but not in mass production?
So that, yes, technically, but it's very hard. Growing animal cells in culture requires, obviously body temperature, 37 degrees Celsius. You need to provide food for the cells. You need to provide protection from disease. You need to provide everything that an animal body provides to its cells. That is food, protection from disease, and cows do it using sunlight. These bioreactors you're going to have to provide electricity to, and depending on the source of that, it is going to affect its kind of sustainability. So yes, but mass production at the level that we produce meat globally is where I think it will not scale to address global climate food concerns, in my opinion.
Correct. Yes, I think so too. Now, here is a question for the Philippines. And this is, how far has the Philippine adopted genetic engineering in its livestock industry? Dr. Marvin?
Okay, thank you very much for the question. So, particularly here at the Livestock Biotechnology Center, there's not much adoption of this advanced modern modern biotechnology techniques. As actually here at the Livestock Biotechnology Center, we are really tapping several experts, local experts here in the Philippines, such as the NIMBB, so as to capacitate some of our researchers in the academe, in the government, so that we will be ready just in case this um, modern biotechnology techniques will be applied at the full scale. So right now, I think we are still in the initial stages or approaches here in the Philippines.
Okay, yes, that's correct. So then the next one would be, I think you can respond, both of you, Alison and Dr. Marvin, with the current repopulation program of the government, maybe we can start to genetically engineer swine that are more resistant to ASF that can be used in the repopulation program. So can we have, yeah, Alison has already put her response. Can you please read, and then we'll listen to what Marvin will say.
Sure, I'd love to. We haven't developed them yet. So lots of groups are working on this, but we have not come up with an approach to produce a genetically engineered or any pig that is resistant to African swine fever. So it's an area of active research, and every time I've answered a question, I've provided a reference link to people. I'm not sure if they can see those, where you can read more about it in open source peer-reviewed papers. But there's work, but nothing that you can use right now.
Yeah. So I guess we won't have anything from the Philippines as yet, because
can I add something on that?
Okay, you do. All right, please.
Yeah, just like Alison said, right now it's still work in progress, but aside from being a researcher, I'm also a field veterinarian, and I've been talking to several farms here in the Philippines. And since it's not yet available, the technology is not yet available. What we really can do right now is to strengthen our biosecurity measures. That would be a very good strategy that we can do right now to really prevent the entry of these diseases in our farm. Because I was able to talk to several farms that still they were able to rear their pigs in some areas here in the Philippines, provided that they have a very good biosecurity measure.
Okay, yeah, that's needed. Okay, so the next one will be from Christian Caldo. What is the probability of having off-target genetic changes when performing CRISPR-Cas gene editing in livestock? Alison?
So you can try to minimize it using very careful guide design. So the little guide RNA that goes in and matches where you want it to cut, you can design that so it doesn't match anywhere else in the genome of interest that you're working on, cattle, whatever. But I think it's important not to get too excited about off-target changes, because that is actually the very basis of all of our genetic improvement programs. So whenever nature introduces an off-target change, that's evolution. And that is actually the basis of evolution and our breeding programs. So there's nothing innately dangerous about an off-target change. And I fear that it's become kind of a boogeyman or a scary thing. And so, yeah, you can minimize it, but if it happens in a food animal, really, does it matter? Because it happens your child has 30 mutations, off-target mutations relative to you, just because DNA repair is not perfect. And so I feel like that has to be kept in context with what occurs spontaneously in natural breeding. And every meal you've ever eaten is genetically distinct and unique from every other meal you've ever eaten. And that in and of itself is not a hazard or a food safety concern.
Yeah, correct. Do you have any other additions, Marvin, please?
No addition.
Okay. Yes, I think we have read all of the ones which were answered. Now we can open new questions. So the first one is, in the Philippines, GM crops has historically been met with resistance, even from legislators. What has been the feedback from current legislators about GM, GE livestock and crops? Are they generally supportive, resistant? How has the DA improved acceptance at the legislature level so products can be commercialized and reach the farmers?
Okay, maybe I will be the one to answer that.
Yes, it is.
Listening to several presentations like Dr. Halos and also Dr. Ola from ISAAA, I think what we really need to okay, at first, our Filipino scientists, by the way, are very our Filipino scientists are very good and very talented, and I know that most of the products that our countrymen can produce are really safe. It's just that we just need to strengthen more on the communication side, which I think ISAAA has a very big role that they are actually doing to continuously promote the good thing that this biotechnology products has to offer to address the progress, and also to be transparent in all of these technologies that we are doing. Because as an example of the GM crops that we have here in the Philippines, slowly but surely, they were able the Filipinos were able to accept these technologies because they can see the positive effect that it can bring to our economy.
Yeah, okay. Those are good answers. Now here we have a question from a scientist from the University of the Philippines, Los Baños, Orville Bondoc. Do we have information on how many of the imported breeding animals and imported milk are products of biotechnology? In the absence of a local genetic improvement program, should we just import and regulate them? How would the application of biotechnology benefit local native animal genetic resources if we don't have a local breeding program for them?
So let me answer that question. So
Yes, please.
I'm not privy of the importation of other government agencies, but so far as Philippine Carabao Center is concerned, so because I was the one who took care of the 2,000 buffaloes from Brazil at that time, and because the genetic improvement program for water buffaloes takes time, so we really need to have we need to do importation, but we have a very good genetic improvement program for that. That's why it's just like starting stock that we can do, and then the rest is to continuously propagate those animals. But for the other livestock species, I cannot answer for that. What is the other question? The milk and meat, I don't have much information on.
Okay, yeah. All right, thank you. I hope Dr. Olvin has been satisfied with your response, but we can talk to him later. Now, one of the very, how do you call this, important issue is the African swine fever, which Alison and Marvin has already discussed. And I think I can put these two questions together. Why is it that ASF has a long-term effect? Piggery owners whose pigs were infected with ASF have to wait for several months before they can take care of pigs again in the same place. And what's happening with the vaccines? If we have it, will it be effective, or what is the limit for genetic engineering to fight against this kind of diseases? So can we have Alison on that first?
I'm really no expert. I think I'm going to give it over to Marvin. He's a veterinarian, and we don't have ASF, and so I'm going to pass it over to him.
Right. Okay.
Okay. Okay. Seeing the condition here in the Philippines, it's actually a multifaceted problem. So even if the the regulatory agencies, are like for example the BAI, were able to contain several um, episodes of outbreaks in certain areas. There is this problem of social problem, I mean, that for example the farmers are really they are very concerned about their loss. So for example, the um, compensation system from the government doesn't work well. So they really need to find ways on how to somehow put back their capital. So even if it is said in the guidelines that you should not transport animals outside the 7-kilometer radius, so they still do it, and that's the reason why the disease is still keeping on proliferating. So it's really a multiple faceted areas wherein all of the stakeholders should be working together so as to completely reduce this problem in the industry.
Okay, thank you. We have a couple of questions on the regulation. And if Alison, would you be familiar with the global scenario on GM animals, GE animals, perhaps? And then we can go for the local scenario.
Sure. So genetically engineered animals, as was alluded to regarding the Cartagena protocol, the presence of novel DNA per se is what triggers everything. So if you introduce a transgene and a novel DNA sequence, that triggers GMO regulation in every country, I think. So where it becomes a little bit more nuanced is with genome editing, where you're inactivating a gene, so just knocking out a couple of base pairs, you're not introducing novel DNA, or you're introducing DNA from one breed into another breed, like the polled gene. In some jurisdictions or economies, that is treated as conventional breeding. Others, like, some jurisdictions like South America, Japan, Canada, others, they treat it as full GMO. And so that examples of that is New Zealand, the European Union, and ironically enough, the United States, if you are using genome editing. So I think the answer to that is nuanced. I will turn it over to the people from the Philippines to answer for your own economy, because I'm not personally familiar with your regulations.
Thank you. Marvin, you can respond with the current regulation and then who are the people involved.
Okay. So as what I mentioned, the JDC is still on the process, right? They are still on the review process. But right now, the Livestock Biotechnology Center is we are conducting several capability building activities, wherein we invited several government agencies like Bureau of Animal Industry, NMIS, NDA, to learn some basic and advanced molecular biotechnology training, so that once this JDC will be approved, just like in plans, our partners from different regulatory agencies will be at least ready for this challenges that we will face. So of course, the Livestock Biotechnology Center, through the coordination with the DA Biotech Program Office, are continuously developing those capability building and other activities that would help our partner agencies.
Yeah. Well, one of the things that come up is the question on food safety and how are you going to help local farmers or local livestock owners or through biotechnology. So we talk about is our guideline looking at food safety as well, and how do we make sure that farmers all over the country can be benefited with biotechnology products. This is for Marvin, Alison. Okay.
Okay, so for the developing of this GM animals, there are some stages that we should follow. And yes, of course, the food safety issue is really a big concern, and that is really one of the important considerations that each researchers or probably other private institutions is dealing with developing these GM animals.
Mhm. Yeah. And then once we have biotech animals, can we send them to all our farmers in the Philippines?
I think
Do
it's
we
too
have
early because
too early to tell?
just what Alison mentioned, in the US there are only two approved GM animals.
That's
So
it. Yes.
as compared to plants, maybe animals is really you really need to pass so many trials before
Yeah.
you can reach those kind of accomplishments from the US.
Okay. Yeah. So there's a question here. I think I can answer this one. Uh, are there initiatives of revisiting the NCBP guidelines? Because right now it is very restrictive in conducting gene editing R&D activities in the Philippines. Do you have anything to say with this? Or maybe Dr. Claro Mingala can clarify? Clary, are you there? Okay, so currently the guideline for the animal biotech is still being reviewed and responded to. So, well, we assure you that our National Committee on Biosafety guideline will be reviewed regularly to make sure that we're addressing all the issues. And it will help also our scientists to develop products of biotechnology. Is biotechnology application to the Philippines more of private sector? So here is a question on, do we have a private sector and a public sector biotech activities and animals?
I think that would be both. Like for example, in the vaccine for ASF, right now we really don't have the capability to do it because it will take time. So as far as I know, the Bureau of Animal Industry and Robina Corporation are doing hand in hand to for the trials of this ASF vaccine, which are still ongoing.
Okay, yes. Okay, here is a very interesting question. As we look at climate change, do we monitor or at least look at the evolution of new diseases and strains? And then do we see that also in bioengineered animals? For example, if we genetically modify or we do gene-edited biotech animal, do we monitor if there are new diseases and strains that come out from that bioengineered animal as well? I think this is a part of the R&D of the biotech animals before they are released to the environment, right?
So maybe I had a previous collaboration with researchers from Spain, and true enough, climate change really affects the how uh, the diseases behave in a certain area, particularly in tropical areas like the Philippines.
Mhm.
Let me just state an example: the fascioliasis. I mean,
Uh-huh.
maybe right now we can see it as a problem in livestock, but we may not know it, but it may affect the humans as well, and also the progression of the disease. Since right now, due to climate change, the precipitation is getting longer, so that is really a good opportunity for the uh, pathogens to really infect more susceptible animals and probably humans. But for the genetically modified animals, I don't maybe I don't have any idea on that, since as what I said, we are still on the initial stage of this endeavor to learn advanced techniques. So maybe Alison can have information.
Yeah, so obviously there's nothing on the market yet, so it's answering in the future. But I will say that we already have selection programs for animals that are resistant to, for example, mastitis in dairy cattle. So that's an inflammation of the mammary gland, and you can use basically the fact that she has a lot of immune cells in her milk as a guide that she's got mastitis, and that can be used as a selection criteria to select for animals that are more resistant, so less likely to get mastitis. That's part of our conventional selection programs. And you could argue, well, maybe that's in the long run going to be deleterious, but it's what we have now to select for animals. And if there's an unintended effect of that, where somehow that makes them more susceptible or the bacteria evolve around it, that's just nature and evolution. And so then we'll try and design a different way to minimize mastitis. I feel like that is certainly with the PRRS resistant pigs that are going through approval for commercialization. That company, which is a large multinational company that can afford to do that, is looking at all of what's called the correlated genetic traits. So if the pig's disease resistant, does it grow as well? Does it get pregnant as easily? Does it have as many piglets? Are there any unwanted genetic correlations? Because that's what animal breeders do all the time. That is part of product development, whether it's genetically modified or not. And so that really is just part of bringing a good quality product to market from a genetic company's perspective.
Yes, I agree with you. It's for animals and crops as well. Now, we don't have any more new questions in. May I ask the ISAAA staff, do we have questions from YouTube? Or we're not in Facebook Live, I guess. Do we have? Send me a message or something. Yeah, if none, I'd like to, so in the meantime, we would like to post the survey, post-webinar survey at the chat box. Now it's already up. So we would like everyone to respond to this post-webinar survey or evaluation form so that we would know if the webinar is very beneficial to you and how we can improve it. And so before we call I think there's another question. Oh, okay. All right, so here is one last question. Since GE without novel combinations will not be covered by the JDC, how will all those GE livestock and crops be regulated? And Dr. Mingala is responding. He's a member of the drafting committee. Can we have Dr. Mingala in the spotlight as well?
Hello, good afternoon. Still good morning. Greetings from the province of Capiz here in central Philippines. I'm sorry I wasn't able to stay, but with regards to the question, it will not be regulated. Definitely, it will not be regulated because if you will consider gene editing without any foreign genes that will be inserted to the animal or to crops or even to fish, it will not be covered by the drafted regulation. So it will be considered as similar to the conventional commodity. Thank you.
Thank you so much. Yes, Alison, we have one person here with a raised hand. He has been raising his hand since the beginning. Mr. Junifer Magkanam, can you please turn on your audio and you can speak if you have allowed to talk. Okay. Mr. Junifer, please. Okay, I think Junifer. Do you want to talk? Okay.
Maybe
No.
he doesn't know
to.
his hand is up.
Okay, so maybe before we end this Q&A, can we hear your last comments or advice or keynotes, keynote messages from your presentation? Something that would steer the minds of our attendees. We start with Alison.
I guess it's nice to see such kind of similar thinking in terms of what the Philippines is thinking, in terms of what science is thinking around how an appropriate regulatory framework might work for these animals. And it was a pleasure being on with Dr. Marvin. I think we are like my animal breeders and livestock people all over the world are very similar. And so I think we see the potential, and, you know, we lose 20% of all animal protein to disease. And here's a tool that could help. And it would be, in my opinion, really criminal not to allow that to happen, because there's a really big opportunity cost with not allowing useful innovations to come to market, especially in livestock breeding. So imagine if we hadn't allowed artificial insemination to take off in the dairy industry, you know, how many more cows would we need to produce the same amount of product? And that kind of opportunity cost is something that's often hidden in these discussions, because it's not obvious what the benefit would be if these technologies are allowed. And I think that's a really important point that I would like to leave with.
Thank you. Thank you, Dr. Alison Van Eenennaam. Now let's have Dr. Marvin Villanueva.
As we can see right now, the Philippines is really facing several problems in the livestock industry, but we should view it as an opportunity. Since what I've said earlier, Filipino scientists, researchers are very good. It's just that we should just give them proper facility, proper support, proper funding. And of course, we cannot do it alone. Collaboration is the key. There are many researchers who are doing a very good job from different areas of the world, and we just need to reach out to them. Those that have similar problems, we can work together so that we were able to bring those technologies that can be applied here in the Philippines. I already we already experienced that here in the PCC as well as in the Livestock Biotechnology Center. Since we really cannot do it all. So we really tap those experts from different fields of expertise, so that we were able to bring those technologies back here in the Philippines, and for the livestock industry to gain those tools that can be used by our country. So I think that's it. Thank you very much.
Yes, those are very important points. So with that, let's give a round of applause, a virtual applause, to our two great speakers. Thank you so much, Dr. Alison and Dr. Marvin. And we now proceed with our closing comments. And this will be from Dr. Ramon Clarete, the Chief of Party of BSAFE Project, Winrock International. Welcome, Dr. Clarete.
Thank you, Dr. Ola. Good morning to all. I'm Ramon Clarete. I'm the Chief of Party of the BSAFE project. So let me introduce the project for those who are just participating in this webinar for the first time. This is BSAFE project is supported by the U.S. Department of Agriculture, a technical assistance program for the benefit of the Department of Agriculture here in the Philippines, and the Food and Drug Administration as well. The objectives of which is to increase productivity as well as trade in agricultural products. The introduction of biotechnologies that would increase productivity is one of the areas of important concern of the BSAFE project. And that's the reason why we partnered with the International Service for the Acquisition of Agricultural Technologies, ISAAA in short, in order to disseminate information about the benefits of modern biotechnology. We have done this, I think this is our fourth webinar with ISAAA. We had done 2 for plants and another one for animal GMO regulations. And this one I just focus on livestock applications. And we will be doing another one also on fish aquaculture applications of animal biotechnologies. This is in support of the government's effort of coming up with a regulation on animal GMOs or GE products. So we look forward to having that regulations. And but in behalf of the organizers, ISAAA and BSAE, we would like to thank our 2 resource persons, Dr. Alison Van Eenennaam from UC Davis. She's the Cooperative Extension Specialist in Animal Genomics and Biotechnology there, and well known all over the world. We're so pleased to have you, Dr. Alison, and for you to share your information about the potential or existing benefits, existing technologies that are available, as well as potential technologies that will be on stream in the next few years. Also, Dr. Marvin Villanueva, our very own from the Philippine Carabao Center. He's the Executive Director of the the Livestock Biotechnology Center and National R&D Coordinator of the Center. For sharing also your time with us and sharing what might be this technology, how this technology can help us overcome our challenges in the livestock industry. And you did mention, I think, a most important challenge for us right now is the African swine fever. For 3 years now, we have been, our agriculture sector has not been able to shoot up its growth because of the drag in growth in the piggery industry. And now we also have another problem with the avian flu. So it's really very important that we make use of technology in order to overcome these problems. But I share with you that it may take some time. And just like Dr. Marvin Villanueva had mentioned, we really need a good surveillance system and make use also of existing technology, one of which would be the portable PCRs that can be used for on-site examination. So we do not have really this kind of approach of almost like depopulating pigs without really very good information about which pigs are sick and which are not. And that's really generating this resistance and from our farmers against depopulation. So anyway, I think we need to support the government's effort to issue the regulations for 2 reasons. The first one is even if we don't have the technologies yet, we can avail of the benefits of the technologies of other countries. Recall the corn in the plant, for example, GM corn. That was the Asian borer. We were able to increase the productivity of our corn by just importing the technologies of other countries and we were able to do that because we issued the regulations. And I think this is the same. We should have regulations as well on animal biotechnologies so that we can avail of the products and technologies of other countries to help us out in overcoming our challenges. Secondly, just like what Dr. Marvin mentioned, we have brilliant and scientists here, but they need to have support in order to conduct research. We are working with the Department of Agriculture, the drafting of the Biotechnology Industry Development Act, BIDA. This is to provide incentives for scientists. There's 2 things here. We can think about this. as encouraging our local scientists, our local research centers to partner with multinational companies who are doing research, and we can contract out, for example, certain business processes in the research processes so that we can continuously develop our innate or native biotechnology industry. So yeah, that's another regulation that Be Safe and ISAAA will be supporting, and that would be the enactment of the Biotechnology Industry Development Act. So two things: let's have our regulations on animal GMOs, and let's ask our Congress to pass the Biotechnology Industry Development Act. With that, I thank you.
Thank you so much, Dr. Claretta. You brought many important things, the challenges that we're having now in our local biotech industry, in the livestock, and also looking forward to the GMO regulation in animals, which will promote the industry, the biotech, the livestock industry through biotechnology. Thank you so much, sir. So with that, we would like to close this webinar. We have reached our target audience, and I guess I hope that you would also respond to the post webinar survey, and this will be your ticket to the certificates that we will be issued. And there are questions about if this is going to be available. Yes, all of the PowerPoint presentations and I think the Zoom recording will also be available, as well as Dr. Alison already also provided us some readings, some materials for further references. There were 3 references that she forwarded, and we will also post that in our ISAAA website. It will be around a day or 2, and you will see all of this there. So thank you so much, and it has been a very fruitful morning for everyone. Let's think about what's happening to the livestock industry and support our government, support our scientists, and of course, support our farmers so that they can benefit from the technology. Thank you so much. Be safe. Thank you so much, USDA, DA Biotech Program Office, Dr. Alison and Dr. Marvin. Thank you. Goodbye. See you in March. We will have aquaculture and fishery webinar, and we will also have a foreign speaker and a local speaker as well. Bye-bye for now, and see you. Have a good day.